All right. Thanks so much. This workshop has been great. Thanks, Nima, for the invitation and for organizing. And all the talks that you just heard in this session were kind of a perfect lead-in. I really liked where we ended in the last talk about the need for developing, I think it's both educational resources as well as really like authoritative open source implementations for a lot of these models, and make it easy to compare these different models to each other. And those comparisons shouldn't just be in terms of like an R-squared score, but they should be matched to some sort of hypothesis that you're looking at in the data, and a kind of user manual for unsupervised learning methods. Anyway, I just wanted to quickly say that at the beginning because my talk is not going to touch upon any of those things. And I think that's really, really important. And there's three data scientists from the Flatiron Institute where I'm half appointed and I'm collaborating with, and we're very much interested in building open source tools in this area. So happy to continue to chat over the reception later. In my 20 minutes, now probably 19 minutes, I'm going to tell you about different work, and it relates to how we think about, or I think about at least, animal-to-animal variability in these large-scale data sets that we've been talking about at this workshop. And you know, I'm a bit of a hybrid neuroscientist, mathematician, machine learner, whatever. But one thing that I really like about this topic is that it turns out to be very interesting and deep mathematically and theoretically. And I won't be able to get into those details today, but we have multiple projects kind of going on in this area under this umbrella of methods that we call shape metrics. And my main goal in this talk is to convince you that there's a lot of practical take-home insights that can be done by wrestling with these with the theory a little bit. So the motivation for the work I'll tell you about starts from a recognition that in many areas of biology and other areas of natural science, scientists often leverage a comparative approach to understand and reverse engineer complex systems. And this is extremely fundamental and important. It's something that you learn in every Bio 101 class. So I'm not going to go, belabor this point too much, but just to say that comparing anatomical structures, for example, across species is absolutely fundamental to our understanding of how the brain is built, and also in neighboring fields like genomics and behavioral science, and on and on and on. In systems neuroscience, I think that this comparative approach is there in kind of edges and corners of the field, but I think it's still nascent and growing. And part of the reason that this is just to say that obviously we are not the first people to think about it, especially in invertebrate systems. There's really nice work being able to leverage the special structure of those systems to compare animal-to-animal variabilities in low-level circuit parameters. And what you find is that, surprise, surprise, animals are very different. But the reason that at a systems level and in the mammalian brain and invertebrate systems, we haven't been able to really address this problem is I don't think that we've had the correct experimental techniques, and we haven't scaled them up in a collaborative manner to begin to ask these approaches with sufficient statistical power. So you've heard about the International Brain Lab. I think that's one initiative that's really exciting in this respect because it's, to my mind, one of the first real examples where we're able to record from many, many mice, in this case 100 mice recorded across 7 different laboratories, all performing the exact same task, and it's a relatively non-trivial behavioral task that they're teaching the mice to learn. So on this slide, you're not seeing neural data, you're seeing behavioral data. And basically, you're seeing learning curves where each gray line is a different mouse. And what you can immediately see is that there's a huge amount of animal-to-animal variability at the behavioral level. So some mice learn the task extremely fast. Those have a steep slope. And other mice, learn the task much more gradually. And if you don't remember anything else from this talk, what I want to at least just put into your mind a little bit and have you go home and think about is, wouldn't it be really nice if we understood what the neural basis of this variability was? If we could have a handle on what is different at the level of representations in visual cortex and other decision-making center brain areas in these different animals, that could help us understand what makes a mouse learn the task fast versus slow, for example. So I'm going to try to tackle that question at the level of what I'll call neural representations, or you can even put in the word neural manifold here if that's more familiar terminology to you. So the purpose of this slide is just to make it really concrete what I mean by a neural representation. And there's obviously many levels and ways that you can make this more sophisticated, but for much of the work that I have done in my group over the last year or so, and for the purposes of this talk, I'm going to keep it as simple as possible. So we're just going to think about a neural network or a biological system that we make a recording from as an input-output device. So if we have two animals or two neural networks, I'm just going to treat them as functions, fx and fy. And the idea is how similar are these two functions? So when you're comparing similarity between two functions, you need to test them. You need to put in a set of probe inputs and then view what the outputs are. So I'll call the inputs u1 through um, and just to be very concrete, it is maybe helpful to think about this as a visual coding experiment. So these different probe inputs are different natural images, let's say, that we present either to an artificial neural network or to a biological system. And in each case, we're going to record a high-dimensional response from these systems, which you can think of as like the average firing rate or the average spike count. Okay, so this is a very general framework. It applies to, you can think about it in motor systems and other sensory systems. We're just looking at kind of sensory input-output responses. And the data sets that you get from this, Martin had a really nice, snazzy visualization for this of neurons versus images. This is the exact matrix here that I'm showing. So this is the output that you would get from a single animal is the idea. Each row is the response to a different input, and each column you can think of it as a neuron's tuning curve across all the stimulus conditions that you presented to the network. Okay, and as you saw in the last talk and many other talks at this workshop, it's really useful to think about these data sets not just as a giant matrix that is not so easy to interpret visually, but to think about this from a geometric perspective. So what I want you to think about is each row of this matrix as being a labeled point in some high-dimensional space, n-dimensional space. I can only draw it in 3 dimensions, but actually all of the methods that I'll tell you about rely on no dimensionality reduction at all. So we're going to compare representations in the full n-dimensional space and remain agnostic to what is like the minimal number of intrinsic dimensions we need to project things down. Although that's a very interesting question on its own right and probably has interesting connections. Okay, so we're going to leverage this geometric perspective in a few slides. And remember the original question. The original question is I'm going to record this in, let's say, 2 animals, and I want you to give me a method that tells me how similar these 2 networks are to each other. And the reason this is not a trivial problem is that we never record the same neurons in 2 different animals. And in fact, I would say that those, there's no such thing as the same exact neuron in a complex mammalian brain that, has billions and billions of cells that have their own developmental history. Like nothing is going to be perfectly matched, unlike in certain invertebrate systems like Drosophila, where you can make this one-to-one match. Okay, so neurons are not one-to-one matched, so I can't just measure the raw distance between these matrices. And people have come up with a number of workarounds to this problem. And this was one of the reasons I got interested in this problem, was looking around the literature and seeing that people were looking at this from many different perspectives. So for example, one way to ask or answer the question, how similar is network X to Y, is to perform a linear regression and use the R-squared score to tell you how similar X is to Y. Other groups in the field do things like compare similarity matrices. So this is the basis of representational similarity analysis, which is very popular in the fMRI community. In the deep learning community, the most popular method is called centered kernel alignment. This ends up being extremely similar to RSA. And also people in neuroscience and in the deep learning community, other people tend to use things like canonical correlations analysis, or CCA. So there's a lot of different methods here, and part of what the goal of my work has been to unify as many of these methods as possible and kind of ask, what do we want out of these methods, and can we see them as all like special cases of a single idea? So what I really want to emphasize is that all of those methods I just showed you are reasonable. I think they're all reasonable to answer the simple question of, if you give me two networks, give me just a quantitative score that tells me, are they more similar than you would expect by chance, for example. So you could create some null distribution and say the similarity is 0.8 and it's greater than you would expect by chance, and you could publish that, and that would be fine. The much more interesting and difficult question that I'm interested in is, how do you extend this when you don't just have two networks that you want to compare, but you have many, many networks, all of which are somehow similar to each other? Like, for example, 100 mice in the International Brain Lab task. All mice, you're recorded in the same quote-unquote brain area, even though the targeting might be slightly different. What can we do with data sets like this? And this is no longer just how similar is mouse 37 to mouse 64. That's not in and of itself an interesting question to ask. The more interesting question is to ask, can we find clusters of animals with similar kinds of neural representations, for example? Can we define a centroid of each one of those clusters? So intuitively that involves like averaging across neural representations in this high dimensional space. Can we do prediction and supervised learning on data sets like this? So if I hold out a mouse, and can I predict that mouse's behavior just by looking at its neural representation? And further, can I do some visualization, not just visualization of a single mouse's manifold or dynamics, but do a visualization of this full cohort of 100 mice? And the methods that I'll tell you about briefly are an approach to answer all of these questions. So here I'm showing you a teaser of a result where each point is not an animal, but each point is neurons from a different brain region in the Allen Brain Observatory. And I've colored the points by their color in the anatomical atlas, but 2 points are in this low-dimensional space are close together based on their functional similarity, based on their tuning curve similarity, more or less, again, using the methods that I'll tell you about. So this is a really nice demonstration. If you look close at this, there's all sorts of interpretable microstructure here, like the visual thalamic areas cluster together. The cortical areas and teal cluster together, and the primary visual cortex separates from other areas of cortex. So I don't want to go too deep into those and overinterpret this plot, but this is just a teaser for the methods that I'll tell you about. Okay, so how do we construct plots like that? Taking a step back, again, this is all built on top of how do we compare neural representations across two experiments. So network X and network Y are two sets of high-dimensional tuning curves, and what I'll argue is that we want a notion of distance between these tuning curves that satisfies the following three axioms. The first is that I want the distance between X and Y to be 0 if and only if X and Y are equivalent by some rigorously pre-specified condition. And I'll mention that equivalence condition a little bit in a few slides. The second thing that I want is that the distance is symmetric, so that the distance from X to Y is the same as the distance from Y to X. This is something that actually I imagine will probably create a little bit of discussion, because linear regression is a method that does not satisfy this. If you run a linear regression to predict Y from X, you'll get a different R squared than if you predict X from Y. So I think linear regression and predictivity scores are a perfectly good approach to ask certain questions, but I will argue for some of the approaches that I'll be building up, it's useful to have this symmetry property. And then the third property that I would also advocate for, at least for certain kinds of analyses, is to have this distance metric satisfy what mathematicians call the triangle inequality. And this just says that the direct distance between any 2 points, so for example, the distance between X and Z, is always less than the distance if I go through an intermediate point Y. So all three of these axioms agree with our physical intuition of what distance and space is, and mathematicians call such distance functions a metric. So the immediate question is why do we care what the mathematicians think, and why is it useful to have a metric space? And I would like to answer that question by arguing that, at least for certain kinds of analyses, for example, if I want to cluster different subjects, it's really, really helpful to be able to rely on all of those axioms. So, for example, let's think about what happens when the triangle inequality is violated. if the triangle inequality is violated, that basically means that you can get point configurations that are, contradictory like this. So x is really close to y and y is really close to z, but at the same time, x and z are really far apart from each other. And if you think about what this would mean for a clustering algorithm, it's going to get confused because the left configuration of points says that I should cluster x and y together and I should cluster y and z together. But the right configuration of points says I should put x and z into different clusters because they're very far apart. So there's this like transitive property of what a cluster is that breaks down in non-metric spaces. And by satisfying the triangle inequality, we don't need to worry about these contradictions. The second is that the triangle inequality makes it really easy to define a neighborhood around every data point, and this is really useful for supervised analyses. So I want you to think about each point here as being a different recording made in a different animal, and these are all points in some metric space. Because they're in a metric space, so first of all, I can color each point by additional data I have about that animal, whether that animal is a good performer or a bad performer, for example. And then, let's say I run another experiment. I record activity in a new mouse, and I don't look at that mouse's behavior, but I want to predict whether this mouse was good or bad. Well, I can look at all the neighbors. I can draw a circle, essentially, a neighborhood around that mouse in this metric space, and then average the behavioral performance of all of its nearest neighbors. This is called a nearest neighbor regression algorithm. It's extremely simple, and people have theoretically analyzed it to death, but it works it can work extremely well depending on the data set. Okay, so these are the kind of analyses that satisfying a metric space would enable us to unlock as a community. And I think it's a pretty powerful thing that at least for certain kinds of questions and analyses you might want to consider. So again, I'm not going to go too deep into the technical details behind this, but the mathematical framework that we built around this is inspired by work in a field called shape theory or shape analysis, and this has been applied to many other areas of science, such as like comparing similarity in bone structure across different animals with different evolutionary divergences from each other. So a lot of the classic methods for this were all in actual comparing objects in 3D space. We're going to leverage this approach to compare neural representations in high n-dimensional space. I'm at 15 minutes. Oh, maybe I was long on the first slide. Okay, fine. I'll speed up a little bit. So as I mentioned, we're leverage this geometric perspective on the problem of thinking of neural representations as labeled point clouds in this high dimensional space. And it turns out, so because each animal has a different set of neurons, intuitively, this point cloud is going to be randomly oriented with respect to each other because each axis in this space is a different neuron. So because the neurons are not matched, you can think of them as being randomly permuted. The neural manifolds or the point clouds are going to be randomly oriented with respect to each other. So one simple solution turns out to be to find the optimal rotation that aligns these two point clouds or aligns the two manifolds and then compute the root mean squared error. So this is exactly what I'm going to show you again on this slide. Now I'm showing you an example where network X and network Y have kind of lower dimensional manifolds, but the idea is the same. We can rotate them into a common space and then compute the distance within the aligned space. And this turns out to be a notion of distance called the Procrustes distance, and it's not immediately obvious why this is true, but this is both symmetric and it satisfies the triangle inequality. But what if you want a notion of distance that is not just invariant to rotations? You want it to be invariant to more general, maybe say affine transformations. No problem. We can pre-process the data by whitening each neural manifold. This is essentially a multi-dimensional z-scoring operation. And after doing that pre-processing, we can run the same procedure. We fit the optimal rotational alignment, compute the distance in this aligned space. It turns out that this notion of distance is closely related to canonical correlations analysis, although not exactly identical. So I'm going to gloss over some of those details, but this is just to say that this isn't a single method that we're offering up, but we're really trying to trace out the possible space of different representational metrics to compare two network representations. And this can you can go even further. So if you want to compare two networks and look at whether there are individual one-to-one matching of neurons, it turns out that you can identify the optimal permutation to align tuning curves between two networks. And this can also be turned into a metric that satisfies the triangle inequality. And then a rotation is a more general transformation than a permutation. So this is the Procrustes distance that I showed you before. And again, we can get looser and looser notions of equivalence between neural networks. So again, we can construct a method or metric that is similar to canonical correlations analysis. And this would tell us that two representations are similar if they're related by a linear transform. And there's even a nonlinear extension to this. And I don't have time to go into the details, but this is just to say that there's active projects within my group and collaborations with others that are tracing down these different special cases within this overall framework. So it's a really rich and, to me, exciting theoretical framework that's going to lead to nice work from our group, so stay tuned. Very quickly, I'll just tell you what the actual practical workflow is here. Once you accept one of these metrics that you want to use for your analysis, all you have to do is compute this symmetric distance matrix of k by k pairwise distances between all points. So for example, if you have 100 mice from the International Brain Lab, you would create this distance matrix. And then you can actually pass this off to various off-the-shelf tools, like this uses a multidimensional scaling to create this plot. If I have one more minute, I'm going to tell you that we applied this to the International Brain Lab data and did the exact supervised learning task that I told you before. So we embedded mice into this low-dimensional shape space. Each mouse has a level of performance on the IBL task, which you can think of as being related to its psychometric curve. And we asked whether we could predict a held-out mouse's psychometric curve from its position in shape space. And the short answer is that you can, at least better than chance. And this is what example predicted tuning curves look like. I'm trying to be as honest as possible to show you a good case on the left and a bad case on the right. What you can see is even when the model makes a kind of bad prediction of the psychometric curve, I think the limiting factor, at least in the current iteration of the IBL data, is the behavioral data itself. There's just not enough trials, so the error bars on the quote-unquote ground truth black are pretty large. But anyway, I think this is a pretty cool analysis where we're able to predict an animal's behavior just by looking at its neural activity and not fine-tuning anything on the behavioral data from that animal. I don't really have time to talk about interesting extensions of this that deal with trial-to-trial noise, so I'll just quickly point you to a paper that we published on this, and we just submitted a bunch of other papers on this. So anyway, there's a lot of other things that if I've piqued your interest on this, please send me an email, and I'll send you pointers to more ongoing work. With that, I'll take questions.
While the other speakers can come up for the discussion after.
So you mentioned that instead of like trying to do a dimensionality reduction, and then looking into you're trying to see what are the description of this system and this sort of like the full representation.
Yeah, or like quantify distances in the full representational space.
Exactly.
Yeah.
So
yeah.
Now what happens if let's say you look at the same set of recordings, but now you limit yourself to a subset of those recorded neurons.
Yeah, because we're always subsampling neurons.
Exactly.
Yeah. So that's actually that isn't. It's most closely talked about in this paper. Okay. There's not a short answer I can give you, unfortunately. If you record enough neurons, it works. But that's probably the best I can say. We have an actual theory that tells you how your error should improve as you record more and more neurons. So that's
Really simple question. So the way in which you showed these high-dimensional spaces was that each stimulus was a point in this high-dimensional space.
Yes. Yep.
And I would assume that the axes are the individual neurons.
Yes, exactly. Firing rate space.
Very good. Why not the other way around?
Yeah. So that's
the stimuli and the points are the individual neurons, because then you could define an individual neuron based on the loading on each individual stimulus.
Yeah.
Right?
So yes, and we wrote this 9-page paper. Actually, the one on the bottom is exactly on an alternative interpretation of this in the kind of stimulus axis space. So there is a completely different interpretation, but they end up giving you the same number at the end of the day. So there's two dual interpretations.